Hi everyone. I once again welcome you all to my series of lectures, that is Understanding Pharmaceutical Science with Dr. Hariharan. So today we are going to discuss about the immunoglobulin, especially the structure of immunoglobulin. So immunoglobulin we can also call as an antibody. That means this lecture is totally dependent upon the what is meant by an antibody and what is the structure of the antibody. So let us understand one by one. The first one, actually in 1930, Kevin Cabot isolated a fraction from a serum called the gamma globulin. Today this gamma globulin is we generally called as an immunoglobulin, because this gamma globulin fraction which was present in the serum was responsible for the prevention of the various infectious diseases as well as cancer. So this identification by Elvin Cabot has developed an understanding about the major humoral mediated immunity. So if I generally ask what is an antibody? So antibody is an antigen binding protein generally present on the surface of the B lymphocyte cell, and it is secreted by the plasma cell. That is antibodies which binds with antigens and helps in the neutralization process. So antibodies can available in both in free form as well as in membrane bound form. Generally it is in present as a membrane bound form in the beta cells. So what happens is that once the plasma cells gets activated, this B cells, that is the B lymphocyte, this activated plasma B lymphocyte, otherwise called as plasma cell, secretes antibodies. These secreted antibodies circulates in the blood and helps in the elimination of the antigen. Otherwise it helps us in marking them by means of a process called opsonization, and helps in the elimination by the phagocytosis by the macrophages or the natural killer cell. So this antibodies serve as an effector molecule of the humoral mediated immunity. So if you see the characteristics of these antibodies, the antibodies, as I already told, it is protein in nature. It is the gamma globulin fraction present in the blood serum. So since it mediates the humoral mediated immunity, this gamma globulin we call as an immunoglobulin. So it's a membrane bounded form as well as in free form. If it is in membrane bounded form, it confers one antigenic specificity for the B lymphocyte and helps in the proliferation. So once it binds with the antigen, then it causes the proliferation of the B cell and producing a multiple clones and helps in the elimination of the foreign antigen. So these antibodies, that is all the antibodies, whether it is an immunoglobulin G, M or anything, and we have a millions of antibodies, all the antibodies have structural features which has an ability to bind with the antigen and participate in a number of effector mechanism, that is the number of mechanism of elimination of the foreign substance, that is the antigen. But all antibodies have a similar structural characteristics. So this why we have to understand about the structural characteristics of the antibody. So if you see the structure of the antibody, so we can also call the structure of the immunoglobulin. So its basic structure, since it is a gamma globulin fraction present in the serum, it is a peptide 1. It is a polypeptide structure carrying of 4 protein chains as I have shown in the figure. So this is the common structure which is present in almost all the immunoglobulins. It carries 4 chains. Out of these 4 chains, 2 chains are heavy chains. So we call this a heavy chain because it carries a more number of amino acid sequence than the other type of chains, which is called as a light chain, which has a less number of amino acid sequence. So we can the immunoglobulin, that is the antibody, is composed of 4 chains. Out of these 4 chains, 2 chains are heavy chains and the 2 chains are light chains. So this structure, that is the 2 light chains are identical in nature, so which is generally having a molecular weight of about 25,000 daltons. whereas the two identical heavy chains are larger polypeptides and having a molecular weight of around 50,000 kilodalton, 50,000 daltons or more. And how these four chains are held together? These four chains are held together by the disulfide linkage. It carries two types of disulfide linkage. One is intramolecular, that is within the same chain, and another is intermolecular, between the light chains and a heavy chain. So if you see the disulfide linkage, each light chain is bound to a heavy chain by a disulfide bond. And apart from this disulfide bond, it is also held together by a non-covalent interaction which is produced by hydrogen bonds or hydrophobic bonds or the salt linkage. This leads to the heterodimer. So we call this a heterodimer because one is the light chain and another is the heavy chain which is joined together. Similarly, the heterodimers or the dimers are formed between the two identical light chain and between the two identical heavy chain, which leads to the formation of four chains of antibody structure, dimers of the dimers. So all these chains will help in the maintaining the structure of the stable structure of the antibody, and also based on the characteristics of this disulfide linkage, we can classify it into antibody into different classes and as well as subclasses. So if you see the two light chains, intramolecular disulfide linkage are formed as I have shown in the figure. Similarly, the intrasulfide disulfide linkage happened between the heavy chain also. Then also the intermolecular between two heavy chains and the two light chain, between the heavy chain and light chain also formed as I have shown in the figure. So apart from this, the structure carries two types of region. One is called the variable region because it varies in amino acid sequence. Second is called the constant region, which has a constant amino acid sequence. So if you see an amino acid, since the antibody is a γ-globulin which is protein in nature, so the protein is nothing but a sequence of amino acids. So the amino acid contains two functional groups. One is an amino terminal and second is a carboxylic terminal. So based on this, we can classify these variable regions and the constant region. If you see the variable region, the first 110 amino acids of the amino terminal of the light chain and the heavy chain differs greatly, and this difference is because of its specificity with the antigen. So I have shown in the figure that the variable region, how it should look like. So the segments of this highly variable region we can classify. If it is present in light chain, we call as a VL, that is the variable light chain region. If it is present in heavy chain, we can recognize as VH, that is the variable heavy chain region. So this regions is very much specifically binds with the antigen. So its display on the antibodies in the variable region is important for the successful elimination of the humoral mediated immunity. As you can see from the figure, the light chain variable region and the heavy chain variable region, which is present at the amino terminus of the antibody. So this variable region is important for the complementary determinant sites, that is the complementary determinant regions. So if you see the difference in the amino acid sequence present in this variable region, we can also call as Vatio-Bailey region, which is binds complementarily with the epitope of the antigen. So that's why we call this as a complementary determining region. So the sequence, amino acid sequence of the variable region of antibody will be complementary to the variable region of will be complementary to the epitope of the antigen. So it can easily bind and interact, and the successful elimination of this antigen can happen. So these complementary determining regions will be present in the variable region of the both light and heavy chain, and it constitutes the antigen binding site of the antibody molecule. So the second part is the constant region. So this region is constant because it doesn't show any variable in the sequence of the amino acids. So that's why it is also termed as the C region. So if it is present in the light chain, we can terminate as a Cl region, that is the constant light chain region. If it is present in the heavy chain, we can terminate as CH region, the constant heavy chain region. So these constant regions will help in the subtype classification of this antibody. So to study this constant region and the antibody binding region, so how they have identified by means of enzymatic hydrolysis. So if you see the enzymatic hydrolysis of the immunoglobulin, so on the enzymatic digestion of immunoglobulin G with an enzyme papain produces 3 types of fragments. Out of these 3 types of fragments, 2 are identical in nature and 1 was the different. As you can see from the figure, so we have taken an immunoglobulin G, an antibody. So it was digested with papain. We got 3 fragments, 2 are identical, which is called as fragment AB, that is FAB, and 1 is different, that is fragment C, FC. So if you see the fragment AB region, these are identical in nature, generally having a molecular weight of 45,000 Daltons and are binding to the antigen. So that is called as fragment antigen binding. So whereas the FC fragment, it is the constant region having a molecular weight of around 50,000 Daltons and has no antigen binding activity because it is also formed, it is also crystallized while in cold storage, so we call it as fragment of crystallizable also. So based on these characteristics of the structure of both the heavy chain and the light chain, we can classify the bacteria. We can get say basic varieties of the heavy chains and plus light chains. So first we understand the 5 basic varieties of the heavy chain. So if you see the sequence of the amino acid present in the constant region, we can classify into 5 patterns, 5 different types. One is called the mu, second is the delta, third is gamma, epsilon and alpha. So these are the basic classification of the immunoglobulin, and these each 5 different subtypes types can be further classified as subtypes also. This basic classification of the 5 we call as an isotype. So the length of this constant region is approximately same in the certain types of isotypes. If you see, there will be 330 amino acid sequence in the constant region for the delta, gamma and alpha, whereas 440 amino acid sequence in the micron and the epsilon. So based on this, we can classify the immunoglobulins into IgM if you have a constant region of micro, if IgG if the constant region is gamma, if IgA, IA stands for alpha if the constant region is alpha, IgD, that is if the constant region is delta, and IgE if the constant region is an epsilon. This is the basic classification of the immunoglobulin based on the structural characteristics of the constant region. This can be further classified into sub-isotypes because the sub-isotypes of this constant region of heavy chain is produced by the slight difference in the amino acid sequence. So generally the sub-isotypes we can find in alpha and gamma, that is immunoglobulin A and immunoglobulin G. So if you see in humans, there are 2 sub-isotypes of immunoglobulin A, that is alpha 1 heavy chain and alpha 2 heavy chain. So immunoglobulin A can be further classified as immunoglobulin A1 and immunoglobulin A2. And similarly, there are 4 sub-isotypes for the gamma, that is gamma 1, gamma 2, gamma 3 and gamma 4. So immunoglobulin G can be further classified as immunoglobulin G1, G2, G3 and G4. So similar variation we can see in the light chain, both in the constant region as well as in the variable region. So these light chain proteins we also called as Bence-Jones proteins. This was differ from individual to individual, especially in the amino acid sequence, especially in the variable region, which is comprises about 100 to 110 amino acids. So whereas the carboxy terminal sequence are very constant, it has 2 basic sequence. So these 2 basic sequence, one was the kappa and second was the lambda. So in humans, if you can see that 60% of the light chains are kappa in nature and 40% are lambda in nature. So a single antibody molecule either contains a kappa or lambda, but it doesn't contain the both. Similar like immunoglobulin G, A, like heavy chains. This is the basic classification of the immunoglobulins. So we can classify immunoglobulins based on the structure, especially the amino acid sequence of the heavy chain. So I have shown in the table, you can see immunoglobulin G, the heavy chain is gamma, which can have a subclass of gamma 1, gamma 2, gamma 3 and gamma 4, whereas light chain for all the types either a kappa or a lambda. Similarly, immunoglobulin M, the heavy chain is mu, there is no subclass, whereas the light chain is kappa or lambda. Whereas immunoglobulin A, which is an A stands for the alpha, it has two subclass, alpha 1 and alpha 2. Immunoglobulin E stands for epsilon, there is no subclass, and immunoglobulin D and the heavy chain is delta. So this comprises about the various structures as well as based on the structures how we can classify the immunoglobulin. So I thank you very much for your patient listening of understanding about the structure of the antibody, and in the future lectures we can discuss about the different types of antibodies in nature and also the various aspects of these antibodies. So thank you very much.